What about voltage gate sodium channels? As you remember, the initial experiment with Pronase was performing squeeze drive axon in voltage gate sodium channels. So where are the parts of the voltage gated sodium channels that account for the fast inactivation of these channels? So sodium channels have a more complicated structures as we're gonna see in our course. But just give a brief idea, uh, the main polypeptide that forms voltage gated sodium channels is about 4 times bigger than each subunit of voltage gated potassium channels. So a single polypeptide actually is equivalent to as 4 subunits of these potassium channels coming together. And as you can imagine, uh, many people try to do similar experiment as what was done with shaker potassium channels, and they start to delete amino acids in the amino terminals, in the C terminals, and so on, and try to find some area of these sodium channels that would be responsible for the fast inactivation of these sodium channels. And what we learned from this experiment is that it's not the amino terminals that's responsible for this inactivation of the sodium channels, but instead it is a loop between domain 3 and domain 4, okay? And this loop here, this intracellular loop, seems to be what's equivalent to the ball and chain domain of the shaker potassium channels. And why is that? Is because when there is amino acid changes in this domain, uh, the investigators were able to remove the fast inactivate of these sodium channels. So here are voltage clamp recordings of voltage gate sodium channel expressed in a cell line. And you can see in the wild type sodium channels, these channels activate very quickly and then also inactivate very quickly. Now, some amino acids in this loop between domain 3 and domain 4, when they were mutated, in this case isoleucine 1488 mutated to phenylalanine, and the phenylalanine 1489 mutated to glutamine, we can see that the inactivation process was in large part eliminated. Also, single mutations were able to replicate this experiment, and also some other mutations of this part of the protein were able to even more completely eliminate this fast inactivation process. What, uh, so far we talked about this fast inactivation process, and we call this fast inactivation process N-type inactivation because it involves the amino terminals in the shaker potassium channels, and we gave it the same name for sodium channel as well, although it's not the amino terminals involved in this fast inactivation process. But also, if you look more carefully on how these channels behave, even shaker potassium channels have another inactivation process that occurs in a much slower time scale. So for example, these shaker potassium channels show very fast inactivation. Um, you can remove the amino terminals and now you remove this fast inactivation. But if you look these mutant shaker potassium channels that do not have the N-type inactivation or the ball and chain mechanism type of inactivation, if you look over a time scale of several seconds, not milliseconds, but several seconds, the second type of inactivation, we don't understand very well how that occurs, but there is some experimental evidence that indicates that involve this slow allosteric changes of these proteins in a time scale of several seconds. And so we think that this process that occurs in a very slow time scale involves the extracellular part of this protein, uh, the evidence comes using extracellular blockers for these potassium channels. That's so we can block this slow inactivation. And this slow inactivation has been called C-type inactivation. So now we can think about voltage-dependent gating of ion channels. The inactivation involves the fast inactivation at least being involved by this ball chain mechanism, but overlaying on this any type of inactivation, there is this slower process that we call C-type inactivation. And some of this future has been well defined now using what we call molecular biology and physiology. What about activation? That's another major feature of ion channels, and the activation involves this very fast opening of these channels. So it means these channels go or transition from a closed state to the open state in response to change in transmembrane voltage, and that also must involve some kind of molecular mechanism. This response to change transmembrane voltage is very steep. So if you look at the voltage dependence for some of these potassium channels, you can see that is a very steep voltage dependence because this channel stays closed from minus 100 to about minus 50, and when the cell depolarized by very few millivolts, these channels transition from the closed to open state very quickly, and then most of the channels now are open with very few millivolts of change. So how you can explain this very steep voltage dependence for activation of these voltage dependent channels? Again, if you go back in time and look at some of the electrophysiology experiments that were designed to provide some insights about what's the mechanism for the activation process of ion channels, the first thing that was postulated, it was that built in these channels there must be what we call a voltage sensor, because these channels are able to somehow sense this change in transmembrane voltage, and then in response to this change in transmembrane voltage be able to open the activation gates to allow the permeation of ions. So what would be this voltage sensor? Well, this voltage sensor, as the name implies, has be able to sense transmembrane voltage. So somehow it must be some kind of amino acid that's charged. Could be negatively charged or positively charged, but because it's charged, it must be able to sense a change in transmembrane voltage. Also, this charged amino acid must be inside of those domains of ion channels that are embedded in the plasma membrane, because parts of the protein that are intracellular or extracellular, they will not be able to sense change in transmembrane voltage. Hodgkin and Huxley actually were the first to suggest the existence of this charge residues on these channels that could account for the ability of this channel to sense changes in transmembrane voltage. With the molecular cloning, then people start to look charge residues that will be in the transmembrane domains of these channels that would account for this voltage sensitivity of these channels. And with careful alignment of this sequence of the amino acids, what was found was that there was a transmembrane segment that was common among all the voltage gated channels that was called S4 segment, because it was the fourth transmembrane segment that had very highly positively charged amino acids. So you can see here in this cartoon that S4 domain is found in voltage gated sodium and calcium channels in each one of those repeats. Okay, 1, 2, 3, and 4. And also the S4 domain is found in voltage gated potassium channels. And these S4 domains are very characteristic because have these positively charged amino acids. So if you look the sequence of S4 region or S4 domain of a shared potassium channel, you can see that the existence of these positively charged amino acids, in this case arginine, 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 lysine, arginine, lysine, that repeats at every 4 residues. And then between this positively charged amino acid, there is these hydrophobic amino acids that allow this region of the protein to be embedded in the plasma membrane. So this part of what we call S4 domain contain most of the charged amino acids in the transmembrane domains, and because of the position of these amino acids, people speculate that this would be the voltage sensor domain of these ion channels. How these ion voltage sensing domains could be detected? First, if this voltage sensing domain is formed by this positively charged or amino acids, then with a change in transmembrane voltage, these amino acids should be able to move across the plasma membrane. So for example, if the cell depolarizes, these amino acids will move towards the extracellular domain of these ion channels. If there is a movement of charge across the electric field, that's equivalent to a current. So many years ago, people postulate the existence of what we call gating currents generated by the movement of discharged amino acids of the voltage sensor domain of these ion channels. So with very careful experiments, actually some investigators were able to measure these gating currents. So the first thing to do to measure those gating currents is to remove the ionic currents, so the currents generated by the movement of ions through these ion channels. So for example, if you are measuring gating currents for sodium channels, you could block the ionic currents using tetrodotoxin. So if you block the ionic currents of tetrodotoxin, and then now you depolarize or hyperpolarize these axons, and you measure what kind of currents you find these axons, you can find these very tiny currents, okay, that we call gating currents. And these much larger currents here will be the ionic currents. And here is an example of a family of gating currents from when the cell is depolarized to -70, to -50, -30, -10, and so on. Okay, and that this would represent the movement of charges of this voltage sensing domain of these channels when the cell is depolarized. Now when the cell is repolarized or goes back to the original resting membrane potential, then the gating currents would move in the opposite way and generate currents that have the opposite orientation. So what are the predictions on neutralizing the positive charge in the voltage sensor? Let's say you have access to a shared potassium channel or a voltage gated sodium channel clone, and now you have the ability to change one by one some of this positive charge in the voltage sensor, and then express these mutant channels, for example, in a cell line, and then measure some of this gating currents and ionic currents. So describe the predict changes on the voltage dependence and magnitude of ionic currents. Also describe the predict change on the voltage dependence and magnitude of the gating currents.